All right, good morning, everybody. Hope everyone's doing okay on this Monday morning. Welcome back to class. We had a little bit of a long weekend from human anatomy over the weekend, and we're going to get back to it today, continuing on the nervous system. So here's what today's class looks like. Oops, get this moving here. We're going to talk about the support cells of the nervous system, of both the peripheral and central nervous system. And then I have a human anatomy in the news, kind of cool story. I think I promised you the story on Wednesday and we never got to it on Wednesday, and we didn't obviously get to it on Friday. So we'll get to it today. And then we're going to continue talking about the support cells and get into neuronal integration, reflex arcs, and things like that. And then we'll wrap things up with a video about how the nervous system is electric and how you can actually control it with external impulses. And I've just got some scheduling reminders for you and some updates, and then we'll wrap things up with Tick It Out. As we move through the material, don't forget you have a study guide to go with all of this, which has useful diagrams for you. You can scan that code there to take you to the Google Slides, where you can download that or save it to your own files and modify it accordingly. So, and I did I meant to take the class program down for today, and I forgot to take it down and I left it up. So the class program for Wednesday is going to be the same one that I had originally planned for today. So if you did that over the weekend, you're a step ahead, you're good. If not, you can see your class program for Wednesday. It just has some reading and a video and a few reminders on there. So with that in mind, before we dive into it, what questions do you have? All right, let's do it. So nervous tissue, just as some reminders, just a little bit of review here. Nervous tissue has two main cell types, remember, and they are very densely packed together and they're intertwined with each other. We have neurons, which are these highly specialized cells that are really good at one thing and one thing only, and that is conducting electrical impulses called action potentials to send signals. And then we have support cells. In the central nervous system, we call these neuroglial cells, or neuroglia. And these are nervous system tissue cells, but they're not the cells that actually conduct the information. These are all of the variety of support cells that keep your neurons healthy and keep them basically running smoothly. So today we're going to get into that in a little more detail. Let me see here. I don't want to go back to that slide. So just as a reminder, we have these two types of cells. Neurons we talked about last week in class. Just as a quick reminder, we had this general schematic of the neuron. We saw the cell body that contains the nucleus, this long axon down which travels this action potential, and then it ends in these terminal boutons where neurotransmitters are secreted. We talked about that last week in detail. Today, we're going to move on and talk about the support cells around these neurons. Go ahead.
I just had a quick question. I can't remember the overall cell structure. Do you know the axon hillock?
Yes, the axon hillock is the spot in the cell. So it's right between that cell body and the axon. And that is important because that's where the action potential originates. Now, we're not going to get into the physiology of action potentials in this class because it's more of a physiology thing than an anatomy thing. But I do want you to know that spot on the cell for sure and why it's important. So the action potential initiates here and then is sent down that axon. Remember, importantly, that axons are one-way streets, that these neuronal cells only send messages in one direction. Now, of course, you have lots of these neurons, which are all firing information in different directions back and forth, but each individual axon is only sending information in one way. It's either sending it from the central nervous system out to either a muscle or a gland to tell it what to do. So those are your motor nerves, or it's sending information back to the central nervous system from the peripheral nervous system, and those are your sensory nerves. Good question. Others? All right, so let's talk about these support cells. As I mentioned, the support cells in the central nervous system are called neuroglia or glial cells in the CNS, and they provide a number of important functions to keep those neurons running smoothly. So one of the things that they do is they cover what we're going to call the non-synaptic parts of the neurons. So basically what they do is provide an insulation. So, you know, when you have an electrical wire in your house and it's like a copper or a metal wire of some sort, and it's wrapped in usually some kind of plastic these days, that provides insulation to keep that electrical signal running where it should be and not bouncing around all over the place. It is the exact same principles, like literally the exact same principles here in those neurons. We have these glial cells that wrap around and keep those axons insulated to keep that electrical impulse firing where it needs to go, and also to keep it moving smoothly and efficiently. Most of the glial cells have these branches, these different processes, and they have a cell body like a neuron, but they are generally smaller. So they have a kind of similar structure in some ways, and we'll see what examples of that in just a second here. But I want you to know that they have cell bodies and they have processes, although their processes take on a different function compared to neurons. Now, what's interesting is that even though the neurons are kind of the famous cells that get all the credit because they're transmitting the information, they're actually outnumbered by about 10 to 1 by glial cells in the central nervous system. So when we think about the mass of nervous tissue, about 90% of it or so is actually these non-excitable cells called glial cells that are supporting those neurons. And so, oh, actually, this is my next bit here. In contrast to neurons, so remember we talked about how neurons were amitotic, meaning that they don't perform cell division. They don't divide over the course of your lifetime. So they're really long lived. They last with you your entire life, but they don't divide and make new cells. In contrast, glial cells do. And this, because they're very numerous and because they do divide and replicate throughout your life, it turns out that most of the time when we hear about something, hear about brain cancer or cancers of the nervous tissue, that is in these glial cells, not in the neurons themselves, because they're more actively dividing, which of course cancer, a simple definition of cancer is just when cells don't know when to stop dividing and keep reproducing themselves uncontrollably. It's much more common in these glial cells than in the neurons. And it's pretty serious. Brain cancer, as you might guess, is not, how do I want to say this? Well, there's no cancer is good to have, but it's one of the worst cancers to get. The survival rate is pretty low, even relative to other cancers. So in contrast to those neurons, they are dividing and reproducing throughout your life. And it's really important that they do so. So what I want to do now is talk about, there's 6 types of these glial cells. There's 4 in the central nervous system and 3 in the peripheral nervous system. We're going to walk through each of those cell types, where they come from and what they do. General outline of glial cells. How's that sitting for you? Make sense? You could kind of think, you could kind of think of it like a movie, right? And in a movie, like what's a current movie now that has like a famous person in the lead role? I don't know. Give me any. What's that? Batman. Yeah. Who's the new Batman? The guy from Cedric Diggory, right? Or he was in the vampire movies. Anyway, yeah, okay. So let's say that you got this new Batman movie. Like he's the star, right? So he's like the neuron. He's the one that is doing the big stuff. He's throwing the punches or throwing the whatever bat tools he's got. And he gets all the credit. But when you get to a movie, right, at the end of the movie, there's like 5 minutes of credits of rolling through with all these names of all the people that made that movie happen. Couldn't happen without them, even though Robert Pattinson's the big star. Exactly the same way here. All those glial cells are your supporting cells. They're not doing the main thing. They're not getting the Oscar nom. Well, they could get out. They have Oscar categories for lots of different things, I guess. That's a bad example. Forget the Oscar analogy. But they're not getting the media attention, but they're making the behind the screen magic happen. So let's look at some of those glial cells. The first type I want to bring to your attention is called an astrocyte. These are the most abundant type of glial cells in the central nervous system. And they kind of help keep things running smoothly in a general sense. But one of the things they do is they regulate the uptake of these neurotransmitters. So remember, at these synapses, neurotransmitters are released and then picked up by other neurons that are in close contact with those terminal boutons of the presynaptic neuron. Well, those neurotransmitters then float around in the cytoplasm, right? And so these guys, the astrocytes, one of their roles is to pick up those neurotransmitters and make sure that there's not too many or too little of them in circulation. So it kind of helps to basically maintain that. And you can see here how this cell structure is similar to a neuron in that it has this cell body with the nucleus, and then it has all of these processes coming out. In this case, the processes are wrapping around those axons, and in this case, also around some capillaries as well. So astrocytes are the first type of glial cell. I want to say a little more about astrocytes, but I'll wait and get back to it in a second. Yep.
I know some drugs, like alcohol, like that, might be up to the uptake of some neurotransmitters. So are they actually affecting the astrocytes and not the nerve cells themselves?
Good question. I don't know, actually. It depends on the drug. Different drugs affect the way those neurotransmitters and the receptors are bound in different ways. So for example, let's say something like marijuana actually stimulates the receptor for certain neurotransmitters. So it makes you think that you're getting those neurotransmitters when you're not. As for alcohol, I'm not sure exactly the mechanism for alcohol and how it relates to astrocytes. I'd have to look into that a little more, but I will say it depends on the drug. Different drugs act in very different ways in that sense. Yeah, that's a good question. Yeah, go ahead, Jim.
Does a single astrocyte connect to more than one neuron?
Yes, great question. Yes. So these guys have they like how these processes expand out and they connect with a whole bunch of other neurons around them, as well as some of the other tissue in the nervous tissue. For example, here you see blood vessels, capillaries. Yeah, good questions. Good questions. Do drugs affect astrocytes directly? I don't know. I'm thinking about that now. Dan, go ahead. Oops.
So when you're saying that in general are like scaffolds, like what is that even? What's the supportive scaffolding?
So they provide structure for those neurons. So if you look here, these processes basically kind of keep everything in place, right? And so they provide structural stability to this network of neurons. Second type of central nervous system glial cell. So remember, we're on the central nervous system here. And so actually what I'll do here is I'm going to make a list just to keep things extra clear. We're going to have the central nervous system glial cells. We had one was astrocytes. Two is going to be microglia. And these guys are, you can see again here the cell body with these processes, right? And these guys essentially act as immune system cells. And they're actually derived from a lineage of immune system cells, and they go around and they basically clean stuff up that shouldn't be there. So invading microbes, dead tissues or parts, dead cells or parts of dead cells and things like that. There's not too many of them. It's the least abundant type of neuroglia, but they're important. They're kind of like scavengers. You can think of them like that. So they're derived from the kind of blood cell called monocytes that serve similar functions in other parts of the body. So these microglia are immune cells that help clean up the nervous tissue from detritus. Next, we have my favorite, and I don't know why they're my favorite, but these Ependymal cells, put these over here. These guys are important in forming the blood-brain barrier. Ependymal cells. So these guys, they kind of act like epithelial cells in some way, and actually, well, they form this permeable epithelium lining of the central cavity. So what they do is they form the lining between the nervous tissue and the cerebrospinal fluid. When we get to the brain lab and a little more on the central nervous system, you'll see that structure and you'll see how there's certain areas where the cerebrospinal fluid flows. Specifically, it encases your brain and then it goes down that central tube of the spinal cord, right? Lining that are these ependymal cells. And they have these little cilia that wave around and keep that fluid moving to keep it circulating. And they provide this barrier. So they're very tightly packed side by side. And they provide that barrier between the cerebrospinal fluid and the brain itself. Oh, I did something really stupid here. Why did I do that? That's weird. Okay, I put this slide in the wrong place. That's fine. Okay, so 3 is ependymal cells. So they're epithelial. They form this epithelial lining around the central nervous system. Yep.
Do they form a lining anywhere else in the nervous system or only in the brain cavity?
Well, everywhere between the central nervous system and the cerebrospinal fluid. So between the brain and the cerebrospinal fluid that surrounds the brain, both kind of on the superficially, but also deep, you have these pockets of cerebrospinal fluid within your brain. We'll see that in lab. But then also all down your spinal cord, there's the tube that contains, that central canal contains cerebrospinal fluid, and they form the lining of that as well. And some of the order that I'm presenting this is a little wonky. You'll see where those structures are in upcoming lab. Actually, not this week, but next week's lab. We'll get into that. Yeah, go ahead, Dan.
Do these ependymal cells create cerebrospinal fluid?
They don't create. It's actually created by some other specialized cells that we'll get to. You'll see these guys. Good question. And actually, I put this slide in the wrong place. I'm going to skip this one and get to the fourth type of glial cells. These are oligodendrocytes. So these guys have just a few branches. So that whenever you hear the term oligo, O-L-I-G-O, that really means a few, kind of roughly. And so that word, so a good example of a word that's been in the popular press lately is oligarchs, right? That means there's just a few people that have a lot of control over things. In this case, oligodendrocytes, meaning they have just a few branches or processes. And you can see what they look like here. Now, these oligodendrocytes are important because they provide the insulation around the axons of the central nervous system. They provide something called myelin sheath. And that's a very important term. I want to make sure you know that one. Myelin sheath is this insulation around the axons. And so the oligodendrocytes are the primary producers of this. And again, that helps keep those axons insulated and helps keep signals moving down. Now, I mentioned a little bit about insulation in terms of astrocytes. Let me pull that back and just to make things more straightforward and simple. I want you to associate insulation in myelin sheath with these oligodendrocytes. They are the producers of that myelin sheath in the central nervous system and provide that insulation. Let's think of astrocytes more as providing structural support to the different cells of the nervous tissue. And you can see here, each cell body of oligodendrocytes has just a few processes that go out and they form this insulative tissue called myelin sheath. It's like a lipoprotein that doesn't transmit electrical impulses, so it provides insulation to the axons of the neurons. Multiple sclerosis is a good example. So this is a disease that can be quite terrible. And it's an autoimmune disease when immune cells actually attack the myelin produced by the oligodendrocytes and degrade it. And that basically prevents nervous signals from being transmitted effectively and can cause a lot of problems, both in terms of sensory input, but also motor control. If you know anyone that's suffered from multiple sclerosis, you know it can be quite debilitating. And so what it's doing is your body, for some reason that we don't quite understand, is attacking this myelin, thinking that it's an invader and shouldn't be there, and degrading it. Like many actually diseases, the problem is our immune system attacking things that it shouldn't. So that's the 4 types of glial cells in the central nervous system. Just a quick rehash. Remember, astrocytes provide structural support. If you need just a catchphrase to go with each of these, I'd say astrocytes provide structural support. Microglia provide waste removal. Ependymal provide epithelial lining. And oligodendrocytes provide the myelin sheath insulation. So if you wanted just one simple phrase to encapsulate the function of each of those 4 cell types. Ethan, go ahead.
With multiple sclerosis, is it the microglia that attack it? Are those immune cells that attack
Oh,
it,
good
or
question. Are there other cells? Oh gosh, I'm not sure. I would guess, and I'll say that tentatively, that it would be the microglial cells because those are the immune cells of the central nervous system. But I'm not 100% sure on that. Yeah. You're always asking me these questions. I don't know. You got it. Okay. Yeah. Jot down these questions though, and shoot me an email at the end of class, and then I can look it up and get back to you. But that'd be my good guess. Yeah.
So kind of like getting back, like there's the base of the question. Is there other immune cells that have that occur in the nervous system, or is it only micro?
If not exclusively, the microglia are going to be the primary. There might be others, but they would exist in small numbers, and I'm not 100% sure on that either. If there could be other immune cells that get through the blood-brain barrier, let me get back to you on that one too. That's a good question. Good. Yeah, is anyone taking the psychology class? Which psychology class? The neuroscience class that gets into this in much more detail? Yeah, if you guys are really interested in this, take the class with Dr. Vadney. Is that who you're taking it with?
I took it last semester, but yeah, we talked
Yeah.
a lot about
She gets way into this, and this is her area of expertise, much more than mine. So if this is an area you definitely want to know more about, she's the person to talk to. What course number is it?
343. Yeah, I'm talking about it in
Any neuro class with Dr. Have you taken it? Does anyone besides Dr. Vadney teach neuro classes here? I'm not 100%. That's right. Of course, Kira does. Yeah, and she's in the psychology department too. So yeah, take those classes, and they can give you much more detail than I can.
clinical neuro goes over it too, like this stuff, but behavioral neuro is like way into it.
Way into it. Yeah. And this is stuff that for me, as someone who primarily works with like physiology and functional morphology in animals, I'm always like, yeah, the brain does stuff and it tells stuff what to do, but that's not really what's interesting. What's really interesting is how big the limbs are and how that help it move across different substances or whatever. But really, this stuff is pretty cool. I should go back to school and become a neuroscientist. It's too late for that. But talk to Dr. Vadney and Dr. Bailey. They're the experts in these areas. Yep, go ahead.
So in terms of the microglial cells,
Mhm.
we just have to know the defense and then just.
Yeah, I want you to know they're derived from immune cells. They're immune cells, and they're primarily, their role is in cleaning up basically things that shouldn't be there, whether that's old dead nervous system tissue cells or other potential like invaders or things like that. Mhm. Good. All right. So with all this in mind, I have a human anatomy in the news here for you. And this is a really interesting study I found. This came out last year, the year before, maybe it's recent, within the past couple of years. And what they did was they actually monitored the activity of brain cells after a person dies. And they found that some brain cells actually boost up their activity after a person is dead. And that's really fascinating, right? And so what they found is they did something called, they looked at gene expression, right? So they looked at the ways that the genes of different cells were producing proteins. And they found that after about 24 hours, different genes responded differently, meaning that the activity ramped up, but it wasn't like they were just doing their normal thing. They were producing some genes more than others. And specifically kind of normally functioning so-called housekeeping genes were maintained their normal functionality. But some other genes increase their functionality. And let me skip to that right here. It's specifically the genes linked to the glial cells. So all of these structural support cells I just mentioned, they boosted up their activity for up to 24 hours. And at the same time, the neuronal cells reduced their activity after just 12 hours. So in other words, after death, these neurons basically fade out and stop doing stuff. Whereas for 24 hours or so, all of these glial cells really ramp up their activity. And that's pretty surprising that 24 hours after you're dead, all of these cells in your brain are still like cranking out proteins and trying to do stuff. And the thought is that they're likely responding to the injury and inflammation in the brain. So basically, as you're dead, right? So your brain's not getting any oxygen, and that causes a lot of trauma to those cells. So those glial cells are basically trying to do their job as best they can and keep those neurons functioning because they don't know you're dead yet, right? They haven't, no one's given them the message that like, hey, this person's kaput, let's give up the ghost here. That's a good expression in this case. Give up the ghost and quit working. They don't know that. Their job is to keep those neurons healthy and functioning at all costs. So they're ramping up their activity for a time to try to keep that going. Now, of course, it can't last forever because nutrients and oxygen are not getting to those tissues. And after 24 hours or so, all of that tissue is going to die anyway. But for a short time after death, some of these cells really ramp up, trying to save the neurons and maintain their functionality. So I thought this was a really interesting study. Who would think to do something like this? Go ahead, Ethan. Yeah.
Does that kind of mean that like the death isn't caused by like brain death per se, but more of a circulation death?
Well,
At least like, I know there'll be some cases where it's brain death, but like in the majority where you see like this, the brain still wants to try and function.
yeah, and in this case, what's important to note is that it's all of these glial support cells that are continuing their activity. And so we associate life in our sense of self with the neurons, with this neural capacity and this neural crosstalk and things like that. So even though these other cells are active, that's not what we consider to be alive, right? That's our brains are not functioning. They're never going to come back in the sense of the neuronal tissue, right?
But you do see the neuronal cells only reduce activity after the 12 hours.
Mhm. Yeah, so it takes a while for them to completely die down, even though that activity is kind of gradually decreasing. So I don't, yeah, there's ethical and physiological arguments about what is actually the time of death, but it's evident that even after we consider something dead, that lots of stuff is still going on in the cells. And specifically here, those glial cells are trying to keep things functioning, even though it's a losing battle. Yeah. Yeah, it's interesting. And this paper does have this great quote that I really like. It says, the results don't prove that zombies are theoretically possible. So just in case anyone was going to take this and misinterpret it, that you're dead and brain cells are still active might mean they're zombies. The authors were very clear that this is not the case. Go ahead, Rick.
So in the case of like the neuronal cells, I know we talked about like even like a few minutes without oxygen can like damage them, so kind of like they can't like come back. So like how did they then last like 12 hours without oxygen?
Yeah, that's a great question, and I don't know the answer. I'd have to go back to the original study and answer that. My suspicion is that basically they start to lose functionality pretty much within minutes, and that degrades over the course of that 12 hours. But how some of them can keep functioning for that time, I'm not sure the answer to that yet. Go back and look. I did provide a link to this like popular science article as well as the original study in your class notes for our class program for Wednesday. So, yeah, that's a good question. And some of those cells might be able to retain functionality for a longer chunk of time, even as the vast majority of them die off. In which case, your brain doesn't do much good if only, let's say, 5% of the cells are still functioning, right? Your brain's pretty much done at that point. So even though a few can survive, doesn't mean the brain as a whole system can maintain its functionality.
Since it takes a while for them to completely die, does that mean that they're still like firing once you're like officially dead, or is it just
Are
like
they firing
maintaining
neurons?
cell function and activity and stuff internally?
Well, are they firing like action potentials?
Yeah.
I don't think the study, I'd have to go back and look at the study and see, because there's a different, what they looked at was gene expression, which is different than action potentials. It's the cells basically proteins, producing proteins from the, well, it's actually not even proteins. It's actually producing RNA from DNA, and the RNA codes for certain proteins. So they're not actually even measuring the production of proteins. They're measuring the production of the transcriptome, or the RNA that's transcribed from the DNA. So, and that doesn't necessarily equate to the production of proteins, or certainly not action potentials. Yeah, so maybe that's it. Maybe they can still transcribe some of that DNA to produce this RNA, but they don't have enough juice to produce these like high energetic action potentials, which require a lot of energy because you have to shuttle all these protons across the cell membranes to get the gradient going and all that. So that probably fits together with both those questions. Cool, great questions. And I wish I knew more about this stuff. Ask Dr. Badney, see what she thinks about zombies. I'll be curious to know. Cool. All right, so let's go back, and I want to talk now. Let's see here. Hold on one second. Let me just orient myself to what I want to cover. Okay, let's transition now. I want to talk about the two types of support cells in the peripheral nervous system, and these are satellite cells and Schwann cells. Never spell the word satellites. Look at that. Today's the day. So two types of glial cells in the peripheral nervous system now we're moving to are the satellite cells and the Schwann cells. The satellite cells They are the cells that provide structural support and surround the cell bodies in the ganglia. So remember, the ganglia are these places in the peripheral nervous system where we have these groups of cell bodies. We'll look at this specifically. You'll learn about something called the dorsal root ganglia, where the cell bodies of your sensory cells are kind of densely packed together. And the satellite cells there provide some structural support for them. Then we have these other types of cells called Schwann cells. So these perform the same function as the oligodendrocytes in the central nervous system, in that they produce this myelin sheath that wraps around the axons of these neurons in the peripheral nervous system. And so you can see an example of this here. We have satellite cells that are surrounding the cell body, kind of protecting it, providing structural support. And then we have these Schwann cells that produce this myelin around the axons that keep those axon signals running quickly and efficiently. So only 2 types of glial cells in the peripheral nervous system. So we've got 6 glial cells altogether. Be sure you know those, know where they're located, peripheral or central nervous system, and know their general functional role. Yep, go ahead.
In MS, would not affect the Schwann cells?
My understanding is the MS is in the central nervous system. So it does not, I don't think, let me say that delicately because I'm not 100% sure. I don't think it affects the myelin in the peripheral. I think it's limited to central, but I should fact check that. Fact check me on that one. I'm not sure. Cool, good questions. All right, so let's talk a little bit more about nervous tissue and the structures. So I alluded to the myelin sheaths that are produced by oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. I mentioned it's a lipoprotein, right? And it provides this insulating layer surrounding the axons. And myelin sheath specifically performs 3 functions. I mentioned this earlier, but I just want to make this explicit here. The first is it prevents the leakage of electrical current. So it keeps that current moving where it needs to down the axon. It also increases the speed that impulse can travel down the axon. When you take a physiology class, you'll see why that is the case. For our purposes, I just want you to know the general function. It's pretty cool. It provides this mechanism called saltatory conduction. Those of you in 251 probably remember this, where the signal can like jump down the axon. And this helps make this impulse propagation energy efficient as well. So you need less energy to transmit the signal. So it keeps the signal moving where it should, it keeps it moving quickly, and it keeps it moving efficiently. 3 purposes of this myelin sheath. And you can see this diagram didn't come up right here, but this is from the textbook. You can see an example. This is a transverse or cross section of an axon. And you can see these layers of myelin surrounding it to insulate it. I don't want to get into, I'm going to skip this next slide. We don't need to talk about the evolutionary significance of myelin sheath. Yeah, I'll skip that. This is enough information about myelin sheath for today. Any questions on myelin sheath? All right, let's transition then. And I want to talk a little bit about repair of damage to your nervous tissue. Now, as I mentioned, these neurons are amitotic, meaning that they don't reproduce. However, if the axon or part of one of these cells is damaged, in many cases, it can still heal. And these are diagrams from the textbook that walk you through the process. So if the cell body dies, if the cell, the nucleus and associated organelles of the cell body die, that's it. It doesn't get replaced. But if the axon is damaged, it can be regenerated. And you can see kind of a walkthrough of what happens here. You see some damage to the axon. You see these bits of myelin sheath floating around. Parts of the axon are here. The macrophages will come and clean it up. So these are immune cells. And then you get this cool regeneration tube starts to form via the help of these macrophages, these immune cells. It basically forms a tube and parts of the axon can start to regenerate. And then it can essentially reform a solid axon. This takes a while. It's not a quick process, but this is why if you damage a nerve in your body, that it can regenerate and can heal as long as that cell body is still alive. Now, it can be difficult to heal, it can heal imperfectly, and it can take a long time, but it is possible. And that's why damage to the nerves can be so destructive, because it just takes a long time, but it is definitely possible. I'm not going to get into too much detail with this. I just want you to be aware that those axons can be, they can be, I don't want to say replaced, but they can be regenerated after damage. Ethan, you're going to ask me a question. I'm not going to know the answer. I'll tell you right now.
With like nerve like transfers of people like who have injuries, is that kind of like taking the process and making it easier on the body by like making less to regenerate or like attach, like to regrow the nerve essentially?
Yeah, I mean, that makes sense, right? You're basically taking the axons, you're basically bringing in some preformed axons and then helping that they help make those connections. Yeah, but it's it's not a straightforward process. Yep, I'm not sure exactly how nerve replacement in that sense is particularly challenging. So that is what the general structure of your nervous tissue looks like. I want to break this down a little more and talk about, when we talk about nerves, what they look like. So nerves are these cable-like organs in the peripheral nervous system. And what they are these bundles of axons. And so they can include both sensory and motor axons, or axons of both sensory and motor neurons, to say that more clearly. And here's a diagram, again, from the book to look at this structure. And what we have is, if we break it down and work from smallest to biggest, you see an axon wrapped in myelin sheath here. And then you see the myelin sheath is wrapped in a layer of tissue called endoneurium. And then we see these groups of these axons wrapped in the endoneurium, bound in something called a fascicle, that's then wrapped in a kind of connective tissue called perineurium. Then we see these bundles of fascicles, the fascicle bundles bound together and then all wrapped up in another layer of connective tissue around all of that called ependymal. So we have these kind of layers of tubes inside tubes inside tubes. First of all, the axon wrapped in the myelin sheath wrapped in endoneurium. We have bundles of those axons wrapped together in perineurium, and then we have those bundles, those fascicles they're called, wrapped together in the outermost layer of connective tissue called ependymal. Now, this structure should sound a little bit familiar to you when we talked about muscles, right? So just to refresh your memory, we have these muscle fibers that are wrapped in endomysium, and then we have these bundles called fascicles wrapped in perimysium, and then those fascicles bound together in the whole muscle tissue, and that's wrapped in something called epimysium, right? And so that is the exact same structure and terminology we used here, except instead of endo, epi, and perimysium, we have endo, peri, and epineurium. But it's connective tissue, and they have very similar structure. Notice within the nerves as well, we have blood vessels that travel the length of the nerves, of course, supplying nutrients, removing waste, all the good stuff that blood vessels do. Now remember, these nerves, each of these axons is extending, is the process extending from one cell body of one neuron. So I really want to make it clear the distinction between a nerve and a neuron. And a nerve is not equal to a neuron. Those are two different things, although they're very closely related. A nerve is a bundle of axons of a bundle of the axons of a whole bunch of neurons. So just remember, in common parlance, we often use those terms interchangeably, but anatomically they have very different meanings. So I want to just introduce you to that structure. We're going to look at the structure of nerves in a little more detail in lab as well. And we'll see some cross sections of slides, and you'll actually be able to see what this looks like under the microscope and everything.
So all those endoneuriums look different neurons?
Yeah, all of the endoneuriums wrap the individual neurons. Yep. So it's a lot of connective tissue in there. And that just adds like structural integrity as well as protection for these nerves, because you don't want them to get damaged, for sure. And you're constantly like, think about the nerves that extend down your arms. You're constantly moving and bending your arms. You want to have a lot of flexibility in those nerves, while at the same time, you We want to make sure they're really well protected. And there's one nerve, the ulnar nerve is actually the one that goes right here. I want to make sure I get to that video today. The ulnar nerve goes right here, and when you bang it, that's what you call hitting your funny bone, and it sends that tingling sensation down because you just messed up all of those axons in that one nerve. Go ahead.
So, when we go and look at the cat, we look at a
Mhm.
is this is this the stuff in like those that have like that skin type color?
Yep, and that's what you're looking, there is the epineurium wrapping up that nerve bundle. That's exactly what you see there. Mhm. Like that lighter kind of fleshy color.
Yeah.
Yep, that's exactly what it is. And we're going to get to that with our cats later in the semester. Let me see what I'm going to say here. Sound good? All right, what time we got? I think I might save our video for Wednesday. But I do want to make sure we have time for some announcement stuff. I'm going to cover a few more slides and then we'll save the video for Wednesday. That'll actually work out well. Maybe I'll even save it for Friday after your quiz. Then you'll just have taken your quiz and you'll be like, I don't want to learn. I want to watch a video. Maybe I'll do that. Okay, so anyway, let's talk about two types of nervous tissue in the central nervous system. So we're moving back to the central nervous system. Remember, that is the brain and spinal cord. And there's two main types of nervous tissue, this gray matter and the white matter. And they're so-called the gray and the white matter because when you look at them, they are kind of dark gray and kind of whitish light gray, right? And so they're just named after the color. The gray matter is a mixture of cell bodies, dendrites, and non-myelinated neurons and neuroglia, right? And so there's lots of synapses in the gray matter, lots of connections being made, as there's some signals being sent via these axons, but generally they're not myelinated. In the central nervous system, the gray matter is usually on the inside. So usually we have the gray matter surrounded by white matter. So here you see a cross section of spinal cord. You see the gray matter here. It's kind of this roughly butterfly or H-shaped tissue here, and it's surrounded by this white matter superficially. In the cerebrum and the cerebellum of the brain, we have some gray matter on the outside as well. And we'll look at that when we get to the cerebral cortex and that kind of thing. So gray matter is the short, non-myelinated interneurons, contains lots of cell bodies, lots of neuroglia. And you can see that here. I want to point out here too, the central cavity that goes down the center of the spinal cord. This central cavity is where cerebrospinal fluid flows. So remember, we have those Ependymal cells lining that central cavity, keeping that cerebrospinal fluid moving with their little cilia, doing their job as they should. So that's gray matter. Let me talk a little bit about white matter. I'm just going to pause, make sure you've got these notes. I don't remember, did I include this diagram on the study guide for this? Are these diagrams on there, or did I stop before I got to this? I stopped. It's not on there. Okay. These diagrams are pretty straightforward though. So talk a little bit about white matter then. White matter does not contain cell bodies. And in fact, it contains just axons, mostly myelinated. And that's why it's white. That myelin, that lipoprotein that makes up myelin is like it has a white color. And so that makes this part of the nervous tissue white. And basically it's all of these tracts of nerves running up and down. And we call these either tracts, or they're called, in the case of the brain, commissures, when they connect the left and the right side of the brain. We'll get to that in a little more detail. The takeaway message here is that the white matter contains these myelinated axons of lots and lots of neurons. And of course, it contains neuroglia as well. For example, oligodendrocytes to produce the myelin that surround all of those axons. And that's all I'm going to say about white and gray matter for right now. That's the takeaway message. We have a lab on the brain the week after this week, and you'll see, is it the week or is it in 2 weeks? I think it's the next lab is on the brain. And so you'll see this in a little more detail. I've got some really nice cross sections of human brains. You can see the white and the gray matter. Okay, I think, what time we got here? That clock is a little slow. Yep, I think we're good to stop here. I'm going to stop at neuronal integration. And that is a good spot. That's a good spot start. We'll pick up with neuronal integration on Wednesday. That's actually works out pretty well. So with that in mind, I want to remind you, you have a quiz in class on Friday, and it will cover the nervous system through neuronal integration. So we will not cover neuronal integration on the quiz. Instead, it will cover everything last week and this week. It's actually even less material than I thought it was going to be because we missed a day last week, but it's still a good chunk of information on the nervous system. Bless you. And that actually works out well. It's in class. It's going to be closed book quiz, so you don't have that much material to study. Basically what we've covered since spring break so far. Remember that you have an exam a week from Friday, and that's going to be in Blackboard Open Book on April Fool's Day. We will have that instead of class that day. I will have it open for the entire day as we've done in the past. It seems to work pretty well. And that's going to cover muscles, skull, and the nervous system, everything we cover in the nervous system up through next week, Monday. So lots of material to cover on that exam. Just a reminder, I've had this on your class programs, reminded you that on your course syllabus, I had the time of our final exam wrong. I want to make sure you've got it. It's on that Friday, April 29th from 1 to 3 p.m. I'll provide details about what that's going to look like as we get closer to the date, but that is the slotted final exam time for this class. So make sure you have that time reserved. Questions on upcoming stuff? All right, reminder as well that you have a lab quiz tomorrow. It's a 10-point quiz covering the muscles of the upper limbs. You can expect questions on a cat that'll have dissected and with pins in it. And then you can also expect questions on one or more of the arm models. It's a 10-point quiz. I think what I'm going to set it up to do is do 10 questions again. So they're each just 1 point. I have to think about that. It's either going to be 5 questions or 2 points each, or 10 questions, 1 point each. So just be sure you can identify those muscles. And then lastly, I've got a ticket out for you. And we didn't have class on Friday, so I didn't have a Venn diagram. I have one for today, breakfast. I don't think I've used this one in class before. And reminder, if you have any questions, I have open hours today from noon to 2 p.m. in our lab room. I'll be there and happy to go over anything, especially as you prepare for your lab quiz tomorrow. Otherwise, take a few minutes and answer these questions as best you can without looking at your notes today. It's kind of a test to see what you remember from what we covered in last week's class on the nervous system, and also a nice reminder for you what you should study for your quiz on Friday. I'll pause the recording here, but of course can stick around for any questions.